Good morning, students. Since a very long time, we have not met each other because I was busy with the practical examination of BSc 6th semester. Just day before yesterday, the practical examinations were over, and as usually, we are going to take the deliberations that we almost ended in the last week. You all know what we have discussed in the last week. We discussed about the simple permanent tissues. I took the class on the very first simple permanent tissue, that was Collenchyma. We discussed about the structure, types and functions of Collenchyma. Today, I would like to take up another simple permanent tissue, that is Collenchyma. As far as this Collenchyma is concerned, it is said to be a living mechanical tissue. It is a living mechanical tissue. Means the cells that are present in the Collenchyma are living. Living cells means, yes, these cells, they are usually having what we call as the protoplast. And in this protoplast, there is an active protoplasm present, and this active protoplasm is usually regulating the metabolic processes. Okay, because of the active metabolism that is going on in the cells of Collenchyma, we call the cells of Collenchyma as living cells, and this tissue is considered as a living tissue. It is not just called as a living tissue, it is also said to be a mechanical tissue. Mechanical means any tissue that is providing strength or support to the plant body is referred as mechanical tissue. Okay, there are different kinds of mechanical tissues that are present there in the plants. Maybe the Collenchyma, the other mechanical tissue, even xylem, although it is a water conducting tissue, at the same time it also acts as a mechanical tissue. Why do we consider the Collenchyma, Collenchyma, even xylem as mechanical tissues? The reason is they are providing mechanical support to the plant body. That is why we refer them as the mechanical tissues. Here, Collenchyma is one such tissue. It provides support to the plant. Okay, assume that this is supposed to be a plant. Okay. Here, the Collenchyma is one such tissue. It usually provides elasticity to the plant body. As that when there is a strong wind, it blows over the plant. What usually happens is, yes, the plant usually bends in the direction of the wind. Okay. Once the wind blows stops, again the plant comes back to its original position. It's all because of the presence of a support giving tissue that is there in the plant. It is supposed to be the Collenchyma. And this Collenchyma, it is one such tissue. It is not present everywhere there in the plant body. Suppose if you consider the roots. If you consider the roots, there in the roots, Collenchyma is usually absent. It may be a monocot root or it may be a dicot root. But we do not find the presence of what we call as the Collenchyma. And it is usually present in the leaves of, is present in the leaves of dicot in the stem of dicots. Okay, one may ask the question that where this collenchyma tissue is commonly present. It is very commonly present in the leaves of the dicot plants. Even it is also present in the stem of the dicot plants. It is said that it is usually absent. It is usually absent in the leaves and stem of monocots. Suppose, say for example, this is a dicot stem. If you take the fine section of this dicot stem, and if you observe this stem section under the microscope, how the collenchyma tissue appears. When you look into the collenchyma, the structure of the collenchyma more or less looks like this. Usually, cells of the collenchyma are polygonal in shape. Sometimes they are also spherical, but quite commonly they are polygonal. Look here. This is how the collenchyma cells look. Polygonal means yes, the cells appears to contain many sides. Even they appear to contain many angles. That's why we call them as polygonal. Cells said to be polygonal in shape. Okay. And I told you, cells are living in nature. Living in nature is supposed to be, they are filled with the granular fluid. Yes. This granular fluid is nothing but protoplasm. There is a protoplasm. This is supposed to be the protoplasm. This protoplasm contains the small nucleus. The bristle contains the nucleus. Yes. Could also observe the presence of what you call as the bacteria, means the fluid cavities, which are very commonly present there in the protoplasm of the collenchyma. Here, cells are usually compactly placed. Cells are usually compactly placed. Compactly placed means they are tightly packed. Between the cells, you don't find any space. Look at the cell wall. This is supposed to be the cell wall. And cell wall is very unique in the collenchyma. Even composition of the cell wall is also very unique. If we chemically test the cell wall present in collenchyma, cell wall is made up of two kinds of polysaccharides. One is cellulose, the other one is pectin. Cellulose is the most common cell wall material that is present in the plant cells. Okay. In the very beginning stage of the development of the collenchyma, cell walls are usually thin and they are made up of the first formed walls. This is the very early formed cell wall, first formed cell wall. We call this first formed cell wall as the primary wall. Primary is the first formed wall. This primary wall is made up of what we call as cells. What happens later is, at some selected places, deposition of another cell wall material takes place, that is pectin. The pectin is going to be deposited there in the corners or angles. Look here, these are supposed to be the corners or the angles. Okay. Exactly in the region of corners or angles, the pectin is going to be deposited. And maybe at the other places other than corners or angles, pectin may be deposited or may not be deposited. If it is deposited, it is deposited to the lesser extent. But there is a heavy deposition of this pectin at the angles or corners. As a result of that, cell walls looks like this. Look here. Okay. There is a more thickening in the region of angles or corners. Even here also, the cell walls more or less looks like this. Can I make out? Exactly in the region of corners or angles, pectin is heavily deposited. That's why the cell walls becomes too much thick in the region of angles of the corners. Okay. That's why we always say cell walls there in the Collenchyma are unevenly thickened. Cell walls are unevenly thickened. Unevenly thickened means they are more thick at one place and less thick at the other place. Okay. The wall layer that is going to be produced at the later stage by the deposition of the pectin is usually called as secondary wall. And this secondary wall is usually called as the later formed wall. And this later formed wall is formed because of deposition of the pectin. And this process of deposition of the pectin is usually called as pectinization. We call this process as pectinization. Because of this pectinization, cell walls becomes unevenly thick. And because of having this unevenly thickened walls, this tissue is able to provide support to the plant. That is why we say it is mechanical in function and it is a mechanical tissue. As far as the deposition of the pectin goes, it is not going to be the same as I have explained in all the different plant species. In most of the plant species, it is like this. There are also other plant species where deposition of the pectin process is going to be different. Say for example, here you can observe the deposition of the pectin exactly in the corners or angles. Because of that pectin deposition in the region of angles or corners, We call this kind of collenchyma as angular collenchyma. We call this as angular collenchyma. There are also other type of collenchyma. Look at this. Here, cells are differently shaped. They are not polygonal in shape. Yes. Cells are somewhat spherical in shape. Yes. And these cells are loosely placed. Because of loose placement, you can see the intracellular spaces. You can make out here. These are the spaces present between the cells. We refer these spaces as intercellular spaces. Here, deposition of the pectin takes place surrounding the intercellular spaces. These are the intercellular spaces. Okay. Just around the intercellular spaces, exactly here, pectin deposition occurs. Exactly here, pectin deposition occurs. There is a more deposition of the pectin exactly against the intracellular spaces. Suppose here there is a intracellular space present. Here what happens? Secondary wall is formed because of this pectinization process. As a result of that, what happens is, yes, there is a wall thickening around the intercellular spaces. Yes. We call this kind of collenchyma as lacunar collenchyma. Because here deposition of the pectin is going to occur exactly in the regions that is around the intercellular spaces. Okay. There is another kind of collenchyma. Okay. Your cells are merely rectangular, just like this. These are supposed to be the cells. Here, deposition of the pectin takes place all along the tangential walls. Suppose if you consider the cell, assume that this is one rectangular shaped cell, just assume. These are the transverse walls that are present in the cell. We refer these transverse walls as tangential walls. And these are the radial walls. Then you can see. Look at this is one radial wall and this is the other radial wall. Okay. These radial walls which are present there in the cells are usually called as the radial walls. Vertical walls are always the radial walls. In this kind of collenchyma, what is happening is all along the tangential, can we calculate? This is the tangential wall. All along this tangential wall, here pectin is going to be deposited. Here pectin is deposited continuously in the form of a plate. Okay. Just like this. When the pectin is deposited in the form of a continuous plate, you can see the thick tangential walls here. This is how the pectin is deposited. When the pectin is deposited, yes, cell wall in this region becomes thick. What I want to tell you is, here in this kind of collenchyma, deposition of the pectin takes place continuously in the form of a plate. That is why we call this kind of collenchyma as lamellar collenchyma. We call this as lamellar collenchyma. Lamellae means plate. Lamellae means plate. Lamella means the collenchyma having the cells with a continuous plate thickening. Whatever may be the kind of collenchyma, it may be the angular collenchyma, it may be the lacular collenchyma, or even lamellar collenchyma. But always these kinds of collenchyma, whenever they are present in the plants, they offer mechanical strength to the plants. Okay, this is really the reference to that of the structure of collenchyma. Okay, let me consider the functions of collenchyma. What are the common functions of collenchyma? Usually collenchyma performs some important functions. Say the very first function performed by the collenchyma is collenchyma is said to be a mechanical tissue. Just now I told you it is a mechanical tissue. It usually provides mechanical strength to the plant body. Suppose, say for example, if the plant body wanted to stand erect, remember, if it wanted to stand erect, particularly the stem, if it wants to stand erect, it needs the support, it needs a kind of reinforcement. Here, collenchyma, it acts as a reinforcement tissue, it acts as a reinforcement structure, so that it gives support to the plant, so that the plant body like stem, it can stand erect. Even the collenchyma also offers elasticity to the parts. Just prior to this, I told you, whenever the strong wind blows over the plant, what happens is, okay, the stem, it usually bends in the direction of the wind. It bends in the direction of the wind, but it never breaks, because collenchyma tissue that is present there in the stem, it avoids the breakage of the stem. Okay, once the wind stops again, the stem comes back to its normal position. Okay, this flexibility or elasticity property comes to the plant because of the collenchyma. In addition to this, collenchyma also performs another function. Collenchyma performs photosynthesis. When there are chloroplasts present. Particularly the young stem. When you look into the young stems, they are green in color because the superficial tissues which are present in the young stem usually contain what we call as chloroplasts. Normally, in the dicot stem, collenchyma tissue is present in the region of hypodermis. Okay, it is present in the hypodermis. Hypodermis is the region that is present immediately inside the epidermis. Suppose if you consider this as section of the stem. Okay, usually the stem is covered with the outermost wall layer. Okay, this is the outermost wall layer. And this outermost wall layer is considered as epidermis. Okay. And just inside this epidermis, we find the region. Say for example, this may be the region. Okay. This region present inside the epidermis is usually called as hypodermis. Hypo means inside. It is present inside the dermis. Dermis means the layer. That is epidermis is the layer, outermost layer. Yes. Inside this layer, you can observe the region. We refer it as hypodermis. The region of this hypodermis, one could observe the collenchyma. And remember one thing, that this hypodermis is also a superficial region. Hypodermis is a superficial region, and the light that can easily penetrate into the hypodermis, so that the cells of the hypodermis getting exposed to the sun or sunlight. And abundant light that is been available to the cells of the hypodermis. Suppose in this hypodermis, if collenchyma cells are present, yes, these collenchyma cells, they are getting exposed to the sunlight. As a result of that, they are going to develop what we call as the green colored plastics in them. These green colored plastics are usually called as chloroplasts. By the help of chlorophyll that is present in the chloroplast, collenchyma tissue can carry out photosynthesis. Remember, photosynthesis is a function often performed by the collenchyma when it contains chloroplasts. These are the two important functions performed by collenchyma. We shall stop here, and in the next class I am going to take up another simple permanent tissue, that is slither. Okay, thank you.